Good evening. Welcome to the international edition of Miggs B on TV. I'm speaking to you now from Norwalk, Connecticut, but just recently I was in London, England, where I spoke with Dr. Alfred Vogler, who's the curator of the Charles Darwin Beetle collection, among others, in the Museum of Natural History, or the Natural History Museum in London, England. This vast collection. Dr. Vogler experiments with them. He cuts them up and chops them up and looks for DNA, and
just beetles?
He knows beetles. Well, there's a special room with the Dave Clark Five, but mainly beetles. And it's a fascinating collection, and never seen by the public, really. It's not open to the public. It's not on display, and we were privileged to visit with him. And we're going to go there now and look at the tape and get an exclusive look at this. Welcome to London. I'm with Dr. Alfred Vogler with the Department of Entomology at the Natural History Museum here in London. And well, thank you for allowing us in here. Where actually are we? What is this room?
Yeah, this is the molecular systematics laboratory, and this is part of this museum where we have mostly collections, but we also have some molecular facilities to study molecular evolution of insects. And so, yeah, that's the...
And what do you do with the, we saw a Bunsen burner flame going, and what happens in this lab?
Right, I mean, what we do here is actually we grind up our insects and extract DNA from them, and I just have here a bunch of them just being processed.
Are these from the collection themselves? Are you grinding up valuable specimens?
We try not to. Okay. Sometimes we have to, and sometimes if things are extinct, for instance, then it's our only way of getting DNA is by going to the collection. But these are specifically collected in the field for the study. And these are things we get from all over the world. I mean, these happen to be from Australia, and we have collectors who go out for us and collect them. And what you see here is this is the specimen is already cut up, and some of it has been used to extract the DNA.
How much do you need to extract DNA? A leg?
Yeah, you can go with a leg only, yeah, but usually we take a little more. So there are 3 parts to the insects, like a head, thorax, and an abdomen. We usually take just the middle part, the thorax, and take that to get DNA, and that's usually sufficient. And these collectors, they put this into these vials, and there's a chemical in there, desiccant, that dries up the specimens very quickly in the field, and that preserves the DNA very well.
And what good is the DNA to you? What do you do with it?
Right, I mean, we then get the sequence, and I mean, we have all kind of machinery here to sequence DNA, and that's then being used to study how these beetles are related to each other.
Oh, I see. So it's
about evolution, and we want to know how things are related, and we can reconstruct the evolutionary history of this group by looking at the DNA.
I see. To really connect it to other species or other...
Right, and to each other. So in this case here, these are all very close related species, and they can be, then there are almost no morphological or there's nothing that can tell us really about how they are related, because they all look very similar.
I see.
But the DNA is helping us out there.
Now, are these from Darwin's? These aren't from his collection.
No.
But how were they gathered? How old are those specimens?
Yeah, these are probably 1 or 2 years old. So there's a person who collected them for us.
People go out in the field and literally just...
They're actually from a desert region somewhere in the middle of Australia. They're quite difficult to get. Only found when it's raining, and that happens quite rarely there. And so you have to be at the right spot at the right time. But in this case, we got a good number of samples here.
And I saw a movie once in the Amazon or a documentary where they get up in the trees and they put a big net and then they shake the tree and everything falls into the net.
Right.
Is that a technique?
That's being done here as well, yeah, and not by myself, but there are lots of people doing this actually. And it was partly pioneered by people here at the museum.
Oh.
And yeah, so it's all about the question, how many species of insects are there in the world, and why are there so many? Yeah, that's the other major question. There's no obvious reason. For instance, there's so many species of beetles. There are,
How many do you know about?
I mean, described there are about 350,000 species of beetles now, and this is 1/5 of all known species of everything else. So there are only like 5,000 species of mammals, for instance. So there are many more beetles. And the question is, of course, why are there so many beetles, right? And other insects as well.
Now, what to the average person, many beetles would look alike. What distinguishes one species? What makes one species from another?
I mean, you have usually some morphological differences. For instance, some are darker, lighter. I mean, between the closely related ones, I don't know if it's easy
So it
to
could
see
be
here. So,
coloring.
like, there's coloring. One is mostly white, the other is mostly dark. Things like this. But of course, you have to look a little closer to see differences. Usually, also only species of one, or only individuals of one species can interbreed and produce offspring. That's another criteria how you can know. But it's mostly looking at them and finding distinct differences between species. And it happens so that there are more different ones in the beetles than in any other group, right?
It's
Yeah, and so then the next step would be to get the DNA, and we're grinding them, and would then extract them with some organic chemicals. And then it's still a very tiny amount only we get from a beetle. And the next step would be to make more of this, so to say, and there is a particular procedure to amplify DNA. And that's what we do here in machines like this. And this is essentially a machine where you can make more DNA starting with very little starting material. It's done using enzymes, using proteins that can make more DNA starting with a particular template. And it's essentially what this machine does, it just regulates temperature. So it's fairly low temperature, this enzyme is active and makes more DNA, and then it's heated up, and the DNA, which is a double strand, is split and melted, and then it's cooled down, and the process is repeated. So it goes on and makes like incredible amount of just one particular piece of DNA, and that's what we then take to amplify them.
Now, do you think the day will ever come when DNA can replicate a specimen like Jurassic Park theory that you could
No, I mean,
replicate?
we can only do very tiny pieces, so that's like nowhere near as much as it would need for full dinosaur. And then to put it in the correct order and so on is,
Right.
I mean, virtually impossible. So I'm not too concerned about this. We're very far away.
All right. Well, thank you. We're in the lab, and I guess we're going to look elsewhere, maybe at some collections.
Hi, I'm Martha Stewart, author and syndicated columnist, and I'm watching Miggs B. For me, it's the next best thing to my style of entertaining.
Now, this is part of what we're looking at here is part of the Darwin collection. These are what we're looking at is actual samples collected by Charles Darwin.
Yeah, right. So in his famous voyage with the Beagle, he collected all those, and you can see here the years when it was done, when it was collected, and it follows very nicely the path of the boat here around South America and South Africa and Australia. So
And what is that big mean one in the middle there?
that's a stag beetle. It's just
Is it
one of the
poisonous,
ones.
or-
It's not poisonous. No, it's actually quite harmless. We have similar things also in Connecticut. You can also see in these labels, many of them has handwriting. It's, I don't know if it comes out easily, but yeah, we have also several hundred other specimens collected by him and always signed with Charles Darwin.
So these are extremely valuable, I would guess.
This box just came back from an exhibit in Japan, and it's traveling around a lot, and just people want to see it.
How many did he spent how many years doing this?
Yeah, apparently it was 4 years on this trip. And
how many did he collect altogether?
I'm not sure how many. I mean, he started as a teenager. He started collecting around here in where he lived, which is like south of London, and his collection is still there. The one Beetles he collected then. On the trip, I mean, he's most famous when he studied the Darwin's finches in Galapagos Islands, and this was just part of what he did there, was collecting beetles. These are just the ones he found, apparently, like accidentally.
I hope he didn't find them in a sleeping bag. The stag beetle is pretty... are there any poisonous beetles? I mean, are they...
Yeah, there are, but they're mostly poisonous when you eat them.
Oh.
They wouldn't...
Not attacking.
Not attack you, no.
And how young were you when you knew you wanted to be an entomologist?
Actually, I haven't been an entomologist for very long. I was trained a bacteriologist and did a PhD in bacteriology, and I switched fairly recently, like maybe 5 years ago, to do this.
But your PhD is in what?
Bacteriology, so it's a very unrelated field then. But this is fairly new. I mean, this whole field of molecular systematics didn't exist a few years ago, virtually, and it's all based on these modern molecular techniques that weren't available a few years ago.
Have you ever found a beetle in your office? I mean, a real, just a... I mean...
Not alive, no.
Okay.
At this point of the interview, Miggs is going to stop and have a spot of tea, as the English say.
Ost, cheese, utan some seamless cheese, seamless cheese, seamless utan some.
My name is Brady, and you're watching Miggs B on TV. I went to London to see the sights.
The tube was fun, but the weather bites.
People talk funny and eat fish and chips.
Everything's pierced, including noses, ears, and lips.
So put me on a plane and fly me.
I think I'm hooked on becoming a limey.
It didn't actually rain that much, but it was overcast every single day. Like on the weather map, on the TV weather, they have like sun and like sunshine here or there. It's just varying degrees of cloudiness. My name is Brady, and you're watching Miggs B on TV.
And at 3 in the morning, I was awakened, flashlights in my face, and the police, and thrown in a paddy wagon and sent to jail, had my head shaved, clothes taken away, uniforms, sent to a real prison 50 miles outside of London.
My name is Brady, and you're watching Miggs B on TV.
And we went to some store, and my dad left to look somewhere else, and I was wearing a black trench coat, and he tried to remember me because I was the guy wearing a black trench coat.
And I'm thinking, oh yeah, he's dressed in black, and he's got earrings, and I looked around, and that described, the closest 5,000 people to me. They're all dressed in black, and they're all pierced, and it was just like a frightening moment of like 10,000 Bradys.
My name is Brady,
And you're watching Miggs B
on TV.
Was there a bounty on these? I mean, do you buy these specimens from somebody, like a freelance person that collects these?
Yeah, some of them we need to buy. Sometimes we have like amateur collectors who collaborate with the museum, and they sometimes.
What does a good specimen go for?
Usually we pay them travel. For instance, I have one person in India who goes there for a trip to, say, 2 weeks, and we pay him 1,000 or 2,000 pounds. So it's mostly covering his travel expenses.
Okay, so it's not per specimen.
It's not usually,
necessarily.
no. I mean, sometimes we have to purchase a specimen, but
If it's very rare, I mean,
yeah,
pay a premium for.
not really. Then we use like closely related ones and
Oh.
stay away from major expenses. Yeah, so then that's the first step, and yeah, so we go through all these.
So
We can-
this happens all day long. I mean,
Yeah, that was
constant,
like would be done every day, pretty much on different groups. There are various projects here, and this kind of amount would take about 2 days to extract, and then the amplifying and sequencing would take another altogether maybe a week for this. So and then the analysis of the data is actually also very time consuming. That will take another month or so. Yeah. So we have to think very long about the data that we get and analyze this in very till we then finally get enough information on their relationships and on their evolution.
Are there other labs in the country or world that do this kind of work?
Oh yeah, there are pretty much all the major museums now have similar facilities.
Do you exchange information?
Yeah, oh yeah, quite a lot. This is also getting very competitive lately because there is not a whole lot of money in this business, and very many people are very interested in this. So there's a lot of competition going on, and also certain groups, some of the major groups. If we know their relationships, that's kind of the end of it. So whoever does it first has the answer, and gets the fame.
Right.
Yeah, so we cannot wait really, and that's why we keep going here even on a weekend. And it's usually very labor intensive. The whole.
Do you publish yourself? Do you publish?
Yeah, we publish. That's of course our main goal here, to publish the results, and that's also quite competitive to get in the most appropriate and the best journals. And all our funding depends really on publications. If we're doing well with this, we get more funding in the next round of applications to funding bodies, and it's a self-perpetuating process. Yeah, I think that's all we can see really here.
Okay.
So then this would be done with many of those.
All right. Thank you.
Yeah, I mean, here, this is the collection. It's actually, this beetle collection here in the Natural History Museum is the largest in the world, really, in terms of how many species there are. So I said before, there are 350,000 described species of beetles, half of which we actually have here, and pretty much everything we study in the lab, we can also then look at here. And for instance, just pull out one of these drawers here, just a couple or just a few of these species, some of which we have also analyzed with the molecular lab.
Interesting. I wonder how many species are in this group. God, they're ugly.
Yeah, that's an interesting question, Diana. There are about, in this particular group, there are 1,000 species along. They're worldwide in distribution. And when you look at this here, and if you went through all these drawers, there are actually not too many differences between them. Their overall appearance, they are all very similar. And that's really what we are attempt to study. Why did they differentiate so much, and why are there so many of these species? Although neither of them is like particularly different from the rest.
Now, this is a room full of cabinets. How many? Just like this. Right? Hundreds,
It's like
hundreds upon
there are
hundreds.
Many thousands, yeah.
Yes, if anybody saw the end of Raiders of the Lost Ark, or that scene in the end with the warehouse, it looks like that almost, right? Endless cabinets of just drawers, and it's
Yeah.
amazing.
And we have very many visitors coming internationally to study these specimens, because many of the specimens are nowhere else, in no other collections. So they have to come here. We send out some also to them, on request. But in many cases, it's just easier for them to travel here and look at them.
Now, how are they preserved? Or are they not anything special?
There's nothing special. No, they dry up, and the outer part is like chitin, so it preserves itself,
Oh.
and they dried up. There used to be chemicals used with the collection just to keep pests out, but now we try to get by without chemicals. I mean,
So you don't have a bug problem here?
we have a little bit of it. But, I mean, if you had come in here like a few years ago, it would have smelled really bad, naphthalene and so on. But we try to do it without now. And we're also exchanging some of these quite nice mahogany cabinets to get them. I mean, there's more
those
time. They're tighter, so they don't let any bugs in anymore.
Or out. You don't want them leaving the building.
Yeah, that's another problem, actually. Some of them are really prized as collectors, and we have a security problem here, and some of these amateurs or people who study these beetles, they sometimes take them here.
And that wouldn't be, I guess, difficult to, well, you'd have to get through the glass case. Now, what are these little color, are these temperature control indicators, or what is that?
No, it's much less sophisticated. It just tells you different regions of the world get a different color. And so these here, they happen to be, this green is for North America. And yeah, so this entire row is essentially from that region. And then there are other boxes which occur, like in India, for instance, they would all have a yellow check mark. Now we have a temperature control somewhere here.
Oh,
but,
okay. Or I thought maybe...
Looks that way.
Okay. Or carbon monoxide detector or something. Okay.
Yeah.
Should we... I understand you have some, actually some specimens from Connecticut, so we might take a look at those now.
I'm Patricia Hurst, and I'm on TV with Miggs B.
Well, I'm in Dr. Alfred Vogler's office, and, you know, excuse my appearance, I've come all the way. I just got to London this morning. I haven't shaved or showered, or but we're all the way to London, and it turns out we have a couple of dozen friends from Connecticut right here with us in the office. There's actually, I'm surprised that there's a collection here from Connecticut. Is that right? What is this all about?
Yeah, this is actually an endangered species in Connecticut, and I have been working on this for a while. A few years ago, there was the problem of how much of the genetic diversity in this endangered species is still left out of what was there previously, right? So it's a species that occurs on the beaches. There's only one population left in New England. It's in Martha's Vineyard, where there's a major part of the beach that's not accessible to the public, and in all other areas, it used to be very common, but it's not there anymore. And so the collection here has specimens still from that period when it was still common, and now we want to see how much of the diversity is really lost. And we can only do this by extracting DNA from these specimens out of the collection. And so what you see here, there are like a bunch of them. I had to clip off the head and get the piece of the body to get DNA out. And I mean, if you look at these old records here, this one, for instance, says Rockaway, New York, 13th of August 1903.
1903.
Yeah. And so then they seem to be quite common there.
I always wondered, when they say an endangered species, especially an insect, insects are so tiny and the world is so big, how do they know there isn't 700 trillion of them in a little place, a little tree somewhere, or not a tree, but in a part of the country, a world nobody's found yet?
Right. I mean, initially nobody really knew if it was going extinct and so on, but people noticed it wasn't there as frequently anymore, and then it was very carefully surveyed, and we now know very well there's just this one population left. Actually, that's not quite true. There was one population close to Martha's Vineyard on the mainland was discovered like a couple of years ago, and I actually have a specimen from there too, and we kind of know how it fits into the, I mean, in terms of its genetic differences, how it fits in with the ones in Martha's Vineyard, and also with those that
So sometimes you do discover an unknown population.
Right, but only, I mean, this was now very, very carefully surveyed, every beach and every little island in the Atlantic, so it's not there anymore. There's just this one. And then there are lots of populations actually further south in the Chesapeake Bay, but in between there's just nothing, and that's pretty well established.
What would be the rarest beetle specimen that you're aware of, or that?
Well,
is there one that's?
Yeah, I mean, this used to be a very common species. Just now the habitat is affected, so it just is now fairly rare. But in some places, in Chesapeake Bay, for instance, where it occurs, you still see like hundreds of them. So it's not that rare. There are other beetle species, like out in the collection here, that have been seen only once ever. There's one record in the collection, and that's actually quite a large number. And yeah, we don't know how rare they are. They haven't been surveyed.
But there's only one specimen exist.
Yeah. And so, of course, many of the species have never been seen. Yeah, so there's probably, I mean, I said before, there's 350,000 known, and probably 10 times as many that are not known. So it's many of them may be rare, or many of them haven't been seen.
So you have a job for life. I mean, you're never going to run out of beetles or bugs, right?
That's for sure.
Does that please you, or sometimes does it get a little overwhelming?
I mean, you cannot, of course, know about all of them, but there are many interesting questions, and you can address with these methods. And yeah, you stick to particular problems, such as you look at populations in one species, or you look at a group of species with a particular question in mind. And yeah, we're also interested in the major groups of beetles, of insects, and how they are related. So we know that the ones in the Chesapeake Bay are quite distinct from the ones in the Connecticut River. And the moment this last population in the Connecticut River gets extirpated, it's gone forever, right? And they also occur on these beaches right on the river there, where people like to drive with their cars, and they simply crush them.
Crush them, and
the habitat deteriorates, and it's almost certain that they will go extinct very quickly.
There's one more soon-to-be extinct species in Connecticut, and that's public access talk show hosts. But maybe after I get some sleep, I'll bounce back. Thank you. Came all this way to find, beetles from Connecticut. The priority with me was revisiting the place I was arrested in London when I was 19, and it was actually I was arrested in a mansion owned by the Queen. It was abandoned. It was being renovated, and somebody had told me I could sleep there because I had been kicked out of a hotel and needed a place to sleep. And also, I had tickets to the Beatles, a premiere of the Beatles. The night before I was to be released from prison was the night that I had a ticket for this command performance of the Beatles. I would have met the Beatles and the royal family. This ticket took me 2 years to get, and I went to London with that intent of going to this concert, exclusive concert for charity that's given every year, and the Beatles did it. Anyway, I missed it because I was in jail, and but the other kids in the prison said they would break me out of jail to go to this concert. Because they really, felt bad for me, but I didn't want to risk it.